Welcome to another Ruffin NeuroLab lecture. Today we are going to be talking about health science issues, but really I'm going to be giving you an overview of biology and foundational concepts. I'd like to give honor to the authors of the particular text, which I will be drawing my information from. And so the authors, as you can see here, are Raven, Johnson, Mason, Losos, and Singer. This is the 11th edition. And of course, this is a biology book by McGraw-Hill Education. So where do we start? I always like to start with an outline. We're going to be talking about the definition of life. That's a difficult one. We're going to be talking about the hierarchy of organization within biology. We're going to be talking about the cell theory. We're going to talk about the scientific method. We're going to talk about organ systems of the human body. We're also going to be talking about negative feedback. We're going to discuss macromolecules. And finally, we're going to end with cell structure and the organelles within the cell. So this is not a traditional lecture. I'm going to tell you things and use the references from which they came and draw from the particular information, the necessary information for this particular lecture. So to start off, Raven and Johnson says that life is very difficult to define. And if you think about it, it actually is. How do you know if something is alive? That's really the foundational question that you should be asking yourself as somebody studying biology. But what you'll find is that it's very difficult to define life. In fact, scientists had to come together to create characteristics, more or less, of things that are alive. We will discuss the 7 characteristics shared by living organisms. The first characteristic is cellular organization. All things that are alive are made of one or more cells. They're so tiny that you can't even see them. The second thing, ordered complexity. From small to large, no matter how small you get, even down to the size of the atom, living things are organized. And everything is made of atoms. So there you go. Sensitivity. Now, I'd like you to keep in mind, we are talking about things that are alive, which includes you. So as the student, you should always be trying to think, how does this information apply to me? And if you felt a certain way about that, that describes the next characteristic, sensitivity. All things that are alive are able to respond to stimuli. Makes me think of when I was younger, how my mother, we were in Virginia driving in the back roads, and we saw what appeared to be snakes on the ground. And my mother is from the country, so this was nothing to her. But I am not. I am a city boy, Hartford, Connecticut, born and raised. So when she told me to get out the car, I think they thought the snakes weren't alive. But they were. So I'm glad I didn't get out the car. Again, sensitivity. Animals should be able to respond to stuff. And so the idea in this particular story was to hit the snake with a stick to see if it would move. And that snake moved off. The fourth thing is growth, development, and reproduction. That's a combination of 3 things, but they all fall under the same umbrella. Living things should be able to grow, they should be able to develop, and they should be able to reproduce. The 5th thing, energy utilization. Everything that is alive uses energy. We'll discuss later on in this lecture where this energy comes from. But as for now, everything that is alive utilizes energy. The 6th characteristic is homeostasis, and this is a very important one. All of them are important, but homeostasis is the ability of an animal to maintain a certain consistency within an ever-changing environment. So this is the ability of an animal to stay at a certain temperature or maintain a certain level of blood glucose. And then the last one Number 7 is evolutionary adaptation. Things that are alive are able to change in response to their environment to continue living and surviving. So those are the 7 things or characteristics shared by living organisms. Cellular organization, ordered complexity, sensitivity, growth, development, and reproduction. Remember, that's all in one. Energy utilization, homeostasis, and evolutionary adaptation. So now we know how to kind of define life, but how do we look at life? Biology, as I mentioned, all the way down to the atom, can be studied all the way up to the biosphere. And so scientists have categorized life at certain levels. They broke it into 3 hierarchical levels. So the 3 levels of biological hierarchy are the cellular level. At the cellular level, you will see atoms, molecules, macromolecules, organelles. These are all the things that are within the cell. So it makes sense that it would be the cellular level. If you combine a bunch of cells, start forming tissues, now you enter into another level called the organismal level. And as I suggested, you will have tissues, and those tissues will form organs, and those organs will form organ systems. This is the organismal level. And as you guessed, if you combine different organisms, you will now enter into the next or final level, the population level. Within the population level, we're going to be talking about different species and biological communities, and even how these biological communities interact with the ecosystem and, as a whole, the biosphere. So the 3 levels of biological hierarchy are the cellular level, the organismal level, and the population level. To help drive this point home, we have figures or illustrations. And we know that a picture is worth 1,000 words. So let's get it to the pictures. At the far left, you will see atoms. Oxygen, carbon, hydrogen, nitrogen. Well, these atoms will combine to form molecules. And molecules will combine to form macromolecules. And in this picture, you're actually seeing a familiar one, DNA. Macromolecules will come together to form organelles, and then finally, within the cellular level, those organelles will be formed to make a cell. As you have noticed from left to right, it's a progression where you're constantly building on a form or principle. And that maintains true as you move from the cellular level to the next level, the organismal level. As you combine cells, they form tissues. And as tissues combine, they form organs. And as organs combine, they form organ systems. And as organ systems combine, they form the organism. So the question that you should be asking yourself from this particular slide is the 5 levels within the cellular level. Those are atoms, molecules, macromolecules, organelles, and finally the cell. Similarly, you should be asking yourself, what are the 4 sublevels within the organismal level? 2 of them are over here. You have tissue and organ, and then you have organ system and organism. And then following along with the same theme, what are the sublevels within the population level? Population, species, community, ecosystem, and biosphere. Whew. As a scientist, there's already a lot of things to think about. How do I know exactly what to study? And when I do study a particular problem, as a scientist, it's important to understand that there are different vantage points to that particular problem. And all of these things are continuous. So we come to a theory, the cell theory, an example of continuous research in a particular area, tested over and over, and the hypothesis stood true. It all starts in 1665 with Robert Hooke, a scientist in England. He was using microscopes, and these microscopes, as you know, magnify things. But at that time, it is not the type of magnification that you have now. This was only 30 times. And following after, another scientist, Anton van Leeuwenhoek, he was using microscopes that were able to magnify things 300 times. So within that short period of time, you saw already that there were technological advances with the microscope. He was looking in pond water. He saw that things were moving around in pond water. So, of course, scientists got together and they summarized the findings looking through these microscopes. And they were able to say that living things, everything that is alive, is made of cells. That is the beginning of the cell theory. But they added on later that all cells come from pre-existing cells. So if you were to be asked, what are the two conclusions in the cell theory? You better say that everything is alive that is made of cells, or everything alive is made of cells, and that cells come from pre-existing cells. So now that we have a theory that we can hold our hats on, and we are now considering ourselves scientists, and we've learned about the different levels of investigation, and we also need to know how to approach a situation or approach a certain problem. So there's something called the scientific method. And this is used all the time, not even within science. It's used all the time. You see something and you question it. And in questioning it, you come up with a possible explanation or a hypothesis. And to verify what that hypothesis is or the validity of that hypothesis, you test it. You may have several hypotheses. And as a scientist, you want to make sure that you test each hypothesis separately to eliminate any variables that may be taking place. And either the experiment that you do is either going to support the hypothesis or it's going to reject the hypothesis. And you're going to continue testing it several times so you're finally able to make a prediction based on the previous experiments. That prediction will lead you to your next experiments where you'll be able to test the prediction and confirm it. And as you see here, I include references for all of this material that I'm using. So if you see a figure and a link, that's actually where I got the figure from. This is a simplified version of the scientific method, showing that the scientific method consists of around 6 components. First, you have an observation/question. Then you have a research topic area that you're going to go into based on that observation. You see this is a circular path. Based on that research, you're going to draw a hypothesis, which you can test with an experiment. Then you're going to analyze the data. And based on your analysis, you're going to report your conclusions. And then the process continues over and over and over again. And as scientists, it's important to make sure that the work that you do is quality, because scientists are building on other scientists' work, and that work will eventually go towards humans and impacting human health. So if somebody were to ask you the 6 components of the scientific method, you would say observation/question, research, particular topic, hypothesis, test with an experiment, analyze the data, and report the conclusion. So as health professionals, we're going to be dealing with humans, and we're going to talk about the 12 major organ systems within humans. We're talking about you, so you need to reflect, self-reflect. What are you made of? I'm showing here 3 of the systems. You have the nervous system, made of the brain, spinal cord, and nerves. Of course, there are other things that are not being named in these particular pictures, but for simplicity, they're naming the major things or major structures. You also have the endocrine system, made of the hypothalamus, pituitary, thyroid, thymus, adrenal gland, pancreas, testes for male, ovaries for female. All of these are endocrine glands. The next system is the skeletal system. Shown here is the skull, sternum, pelvis, and the femur. So you have 3 of the 12 major organ systems right now. Let's continue. Number 4 is the muscular system, the pectoralis major, the biceps, the rectus abdominis, the sartorius, quadriceps, the gastrocnemius. These are all muscles in the muscular system. You also have the digestive system, salivary glands, esophagus, liver, stomach, small intestines, large intestines. This is a familiar one to a lot of people that aren't even, that interested in science. The circulatory system. We all have hearts, and we refer to our hearts in February because we say that it's related to love and everything. But the circulatory system consists of more than just the heart. You have veins and the arteries, all of these things pushing blood around the body, pumping blood around the body. So we are now 6. We have the nervous system, the endocrine system, skeletal system, muscular system, digestive system, circulatory system. Let's move to 7, 8, and 9. The respiratory system. This is one of my favorites. Here is shown the trachea and the lungs. You also have the urinary system, made of the kidneys and ureter, and the bladder and the urethra. The integumentary system, hair, skin, and fingernails. So just as a review, we're almost to 12. We have 9 so far. We have the nervous system, the endocrine system, skeletal system, muscular system, digestive system, circulatory system, respiratory system, urinary system, integumentary system. What's the last 3? Well, actually, the last 3 is the last 2. You have the lymphatic immune system, which is the defense system, and here is shown the lymph nodes, thymus, spleen, bone marrow, and lymphatic vessels. But the last 2 are actually the reproductive system. The male counterpart is the vas deferens, the penis and the testes, and the female counterpart is the fallopian tube, ovary, uterus, and the vagina. So to name all 12 major organ systems, you have the nervous system, the endocrine system, skeletal system, the muscular system, the digestive system, the circulatory system, the respiratory system, the urinary system, the integumentary system, the lymphatic/immune system, and the reproductive system, male and female. Whew, I need something to drink. This is a lot of information. I guess I need to digest it. So the next thing I want to do is just give an overview of which each of these systems do. I did mention components of them, but because this is part of you and part of health in general, it's important to review what these systems do. Just a brief overview. The nervous system, as I mentioned, is the brain and the spinal cord and all the nerves that come from it. This includes the sensory organs, which we are very familiar with. These are the reasons that we see. So we're mentioning our eyes, our hear, ears, touch, taste, smell, all of these things that receive information from the external and internal environment and let us know about the world that exists, and gives us the appropriate motor or commands to adjust and respond appropriately. Not only is the nervous system involved in controlling the body, but the endocrine is also involved in controlling the body. The endocrine system uses chemicals. These chemicals, we traditionally think of puberty as, something that happens in growth. Well, this is the endocrine system. It's using chemicals that are released at a certain time, and they cause responses in distant organs traveling through the blood. This is hormones I'm talking about. The musculoskeletal system. This is now a combination of 2 systems. You have muscles, which are used for movement, and you also have the skeletal system. This is the rigid framework that makes up our structure. Well, the muscles are attached to the skeletal system, which is a place that the muscles can actually cause, when they contract, a change in the distance or the change in the movement of a particular thing. You have a wide variety of finely controlled movements. We also have the digestive system. And this digestive system is basically breaking down the food that you ate chemically and mechanically. You're absorbing the nutrients in that food and you're eliminating what is waste. You also have the circulatory system. As I mentioned, this is the heart and the blood vessels. And it's pumping around blood and nutrients that are in the blood, but it's also getting rid of toxins by passing into other organs. The respiratory system, that's the lungs, and the lungs are giving you the oxygen you need to survive and getting rid of the carbon dioxide, a byproduct of cellular respiration. You also have the urinary system. This regulates body volume. It's using the concentration of body fluids in a process called osmoregulation to adjust and be appropriate to the environment. You have the integumentary system. As I mentioned, this is the skin. This prevents bacteria, viruses, all types of things from actually entering into the body if the skin is actually intact. Once within the body, the immune system, another defense system, uses antibodies and other special types of cells to get rid of invading organisms that are viewed as threats in cellular, in the survival of the cell. And sometimes the cell doesn't recognize its own cell. And so this is a system that becomes compromised. And the final system, as I mentioned this too, you have the male and the female reproductive systems. This is where the gametes develop and where the nurturing takes place. And also this has the complementary structures to allow opposite, the male and female, to come together to produce another organism. So that's an overview of each of these systems, and the drawing shows you where those systems are in relation to each other and in the body. But all those systems need to work together because the cell is extremely sophisticated and the body is extremely sophisticated. There needs to be a way to make sure that processes that are supposed to take place are taking place, and that is used or that is maintained by a thing called feedback. Feedback is used to achieve homeostasis, or making sure that the internal environment stays stable as the outside or the external environment changes. So how does this work? There's a stimulus that is detected by a sensor. And that sensor, based on the stimulus, is going to send information to an integrating system. That integrating system is going to compare and give a result based on the comparison to compensate for any deviation that the sensor is detecting. That deviation or the output of the effector is going to be the opposite of the original stimulus. And so that is going to move the system back towards the set point. So I know that was a lot of boxes and information, but it's more realistic when you put it towards one of the things that's being taken place in the body right now. Right now, you're monitoring your temperature. Above a certain temperature, your body acts a certain way. Below a certain temperature, your body acts a certain way. Those responses to those changes in temperature are going to eventually bring the temperature back to the original set point. And that goes or maintains true for pH, concentration of glucose and oxygen, and a whole bunch of other things taking place in your body to make sure that your body is functioning properly. This is called homeostasis. So let's give a good example. First, there's positive and negative feedback, but negative feedback is more often used than positive feedback. In this example, on the left, we're going to look at a thermometer or a temperature controlled house. And on the right, you're going to look at a temperature controlled body. On the left, the house is supposed to be set at 70. And the sensor is monitoring the air. The stimulus is a change in temperature, which is detected by the sensor. And so the sensor sends that information to the thermostat to compare what is the temperature and what do I want it to be. If the temperature is too high, the AC will turn on and the furnace will turn off and the room will cool down to go back to your original set point. If the temperature is too low, the AC will turn off, the furnace will turn on, the room temperature will increase back towards the set point. And in this way, the output on both sides will go back to the sensor to compare to original values where the deviation is. And this is taking place in the body as we speak. With temperature, our body is supposed to be 37 degrees, and we have neurons in the hypothalamus that are monitoring that. When the temperature changes in the body, those neurons in the hypothalamus detect that. And then they send output from the hypothalamus to try to get the body back to that temperature. Now, if the temperature is too high, what happens? Think about it. Your blood vessels dilate in your skin and you start sweating. Your body temperature will drop and the neurons in the hypothalamus will detect that. What if your body temperature is too low? The blood vessels will contract and your muscles will contract and you'll start shivering. The temperature will rise. Both of these are examples of negative feedback to bring the body back to its normal temperature. So we touched about the whole body and we touched about all these systems, but it's important to recognize that the cell, although we have a bunch of different systems, the cell has general features that can be described for each of them. Every cell is made up of 4 macromolecules. And here you're seeing the 4 macromolecules. The 4 types of macromolecules in the human body are carbohydrates, nucleic acids, proteins, and lipids. An example of carbohydrates would be glucose. Examples of nucleic acids would be nucleotides, or the subunits, of carbohydrates would be glucose. Nucleic acids, nucleotides, proteins, subunits would be amino acids, lipids, and as you see, there's a bunch of different types of lipids. Another thing to point out within these particular classes, types of molecules. Carbohydrates, an example would be cellulose or glycogen or starch. Under nucleic acids, DNA, RNA. Proteins, they describe different types of functional proteins and structural proteins. And then lipids, phospholipids, prostaglandins, steroids, terpenes, triglycerides. The function is also given for each of these particular macromolecules and examples. One thing I want to point out is that each of these macromolecules, carbohydrates, proteins, and lipids, excluding nucleic acids, is found on your food label because you are what you eat. So the food label, they have changed the food label at the FDA. This was the old label. They're moving to this newer label that's bringing out some of the important things that really aren't as highlighted on the old label. But I'm going to be highlighting the fats, which are the lipids, carbohydrates, and the proteins. You're actually eating the substrates for your cell to be healthy. It's also mentioned in cholesterol, which is another lipid, sodium, which is minerals, and you also have vitamins down here. And so it's important to read the labels. Well, what are these macromolecules that I'm describing? Carbohydrates, those are sugars. And so all carbohydrates have this ratio of carbon to hydrogen to oxygen at a molar ratio of 1 to 2 to 1. That means you're going to have 1 carbon, 2 hydrogen, and 1 oxygen. multiples of that combination. So let's look at some types of carbohydrates. An example of a 3-carbon sugar would be glyceroldehyde. An example of 5-carbon sugars, well, they've given you 2, ribose and deoxyribose. Hopefully you should recognize that these are the sugars in RNA and DNA. This is actually a sugar that's in phospholipids, as we get to it a little bit later. Examples of 6-carbon sugars: glucose, fructose, and galactose. So that's carbohydrates. 1 down, 3 to go. Nucleic acids. It was not on the label because you get these from your parents. This is carrying the genetic information about you. And it can be in 2 major forms: deoxyribonucleic acid, or DNA, or ribonucleic acid, RNA. And this is the general structure of a nucleotide, where a nucleotide is made up of a sugar. Remember, that's the 5-carbon sugar that we saw previously for carbohydrates. And the difference between DNA and RNA is that this sugar either has an OH, if it's RNA, at this position here for number 2. And if it doesn't have anything, it just has an H, it's the DNA. So deoxy, which means that there's no oxygen there, is why DNA has the deoxy ribose. That's the type of sugar, nucleic acid. So you have a sugar, you have a phosphate group, and then you have a nitrogenous base. So the 2 main varieties of nucleic acids are DNA, or deoxyribonucleic acid, and RNA, ribose nucleic acid. The 3 components of a nucleic acid, hopefully you know now, there's a phosphate group, a sugar, and a nitrogenous base. Looking deeper into nucleic acids, you'll see that the nitrogenous base come in 2 forms. You either have purines, like adenine or guanine, or pyrimidines, like cytosine, which you'll find in DNA and RNA, thymine, which is only in DNA, and uracil, which is only in RNA. And on the picture on the right, you're actually seeing 2 sugar phosphate backbones combining. What makes the sugar phosphate backbone? The sugar connecting to the phosphate, connecting to another sugar, connecting to a phosphate, connecting to another sugar. So this strip right here is actually sugar phosphate, depicted as a strip. In between are the nitrogenous bases from each strand connected. And what I want you to notice here is that A will always connect with T through 2 hydrogen bonds. C will always connect with G through 3 hydrogen bonds. And so this is the perfect love story. A will always find T, C will always find G. So the 2 nitrogenous bases, which are purines, are adenine and guanine. The 2 nitrogenous bases, and actually that's 3 nitrogenous bases, which are pyrimidines, are cytosine, thymine, and uracil. And the type of bond that connects the sugar phosphate backbone of DNA are hydrogen bonds. We have now finished the nucleic acid section, and we are moving on to the protein section. To left, proteins. What are proteins? They're the most diverse group of biological macromolecules. Proteins do everything. And at the health level, I would like you to think that the majority of issues that take place are some type of dysfunction or malfunction of a protein. Now, there are other reasons, maybe genetic reasons, but then again, the genes produce the proteins. So proteins do everything. What is everything? Here's a short list of 7 things or 7 functions of proteins. Proteins are enzymes. There are a bunch of reactions that take place in the body. Proteins help facilitate these reactions by being enzymes that catalyze the reactions. Proteins are also involved in defense. So I mentioned the immune system. Antibodies, they're actually proteins that are used to help fight invading organisms. Transport, proteins can be transport proteins. They can be ion channels. They can be a bunch of different things that allow something to move from one place to another place. Proteins are also involved in support. So the structure of your muscle or the structure of your bone or even the structure of a cell itself is dependent on protein fibers forming as a building has its infrastructure, similar infrastructure of your particular cell. Proteins are also involved in motion. Muscles are made of proteins, and it's the actin and myosin sliding against each other that even causes the contraction that causes the motion of the human body. Proteins are also involved in regulation. So I mentioned hormones and second messenger systems that allow information from outside the cell to get inside the cell. This is all proteins. And the 7th thing, I'm sure there's a lot of things that they could have mentioned, but the 7th final thing is storage. Proteins are used to store like calcium and iron, things that are in the body that need to be stored. So the 7 functions of proteins are enzyme, catalysis, defense, transport, support, motion, regulation, and storage. And if you see in the top right corner, just as a reminder, proteins are made up of amino acids. And so the most general amino acid structure is depicted on the right. If you notice at the top of it, there's an R. That R represents the variable group of the amino acid, which gives the amino acid its particular name and also functional characteristics. So let's look at the R. How many different R's are there? There are 20 different R's. And the purpose of this slide is not to get you to remember all 20 amino acids, although that would be wonderful. But the purpose is to show you that these amino acids actually can be grouped based on properties. The ones that are in the green are nonpolar. They do not like water. The ones that are in blue are polar, and the ones that are in red also are polar. Why does this matter? Because the nonpolar ones do not like to interact with water. The polar ones do like to interact with water and or will interact with water. And the charged ones want to interact with water. Why is this important? Because the cell is in a liquid environment. Inside the cell is liquid, outside the cell is liquid. And now this is showing you which of the amino acids will interact in the liquid environment and which ones will not interact and try to get away from the aqueous environment. Which leads me to lipids. Some things like water and are soluble in water. Some things do not like water and are not soluble in water. Lipids are the class of macromolecules which are insoluble in water. The reason that they are insoluble in water is there are no places for the water to interact with the molecule. This molecule has a high proportion of nonpolar, remember those things don't like water, carbon-hydrogen bonds. So these lipids will have long chains of these carbon-hydrogen bonds called hydrocarbons. And that property alone, the fact that there is no place for the water to bond, the fact that the bonds themselves between the atoms involved are nonpolar, and you have a high high content of hydrocarbon bonds, produces structures like this. These are 3 examples of lipids. Triglycerides, here are these hydrocarbon bonds. Tri means 3, glycerol, aldehyde, which is the sugar. And here are 3 hydrocarbon chains. So this is triglycerides, otherwise known as fatty acids. Now you have saturated fatty acids, which is up here, and unsaturated fatty acids. The difference being these double bonds. If there are hydrogens everywhere there could possibly be in these fatty acid chains, then this is considered saturated. But because here, this double bond can accept additional hydrogens, there are places that hydrogens can actually add to this chain. These are unsaturated. Something that I would like to mention about these double bonds in this hydrocarbon chain is that whenever there is a double bond, it makes the structure go from being a straight linear structure to a bended structure. We'll come back to that as we discuss the 4th example of a lipid. So here's the first example, triglycerides or fatty acids. The second example are terpenes. These are hydrocarbons in plants. You also have steroids, the 3rd example, like cholesterol. And the 4th example are phospholipids. So 3 or 4 examples of lipids are triglycerides, terpenes, steroids, and phospholipids. Phospholipids form the core of all biological membranes, and they are made of 3 components. They're made of glycerol, which is a sugar, glyceraldehyde, fatty acids, and a phosphate group. This phosphate group has a charge on it. And because of that, this area of the phospholipid actually likes water. Remember, the tails do not like water. They are hydrophobic. But the head, or this top part of it, is hydrophilic. The fact that this molecule has both properties, hydrophobic and hydrophilic, means that this molecule actually is amphipathic. It has hydrophobic tails and hydrophilic heads. So in water, as you can imagine, the tails do not like water, so they are going to cluster together and form a structure like this, where the heads are exposed to the water and the tails are isolated from the water. This is called a micelle. The lipid heads are hydrophilic, the tails are hydrophobic. Another structure that can form in water are phospholipid bilayers. Now you have these heads facing the water here, heads facing the water there, and the tails have lined up together to form a barrier that keeps these 2 water areas apart from each other. 2 structures what 2 structures can a phospholipid form spontaneously in water? Micelles, a phospholipid barrier. And we are closing the end of this lecture to show you that all of these things come together to form a cell. This phospholipid bilayer is actually the membrane of the cell, and any organelle is also surrounded by a phospholipid bilayer. The continuation of this lecture will discuss the organelles within the cell and the roles that they play in cell function. And it will also discuss in detail cellular respiration, which is taking place in the cytoplasm and also in the mitochondria to produce the body's form of energy, ATP. But just as a brief overview, DNA is found in the nucleus. So a lot of that information that we were discussing, I encourage you to continue looking up through scientific journals and other resources. Within the mitochondria, you're making ATP, the body's source of energy. The plasma membrane is the organelle that surrounds the entire cell. And even ribosomes is where, or the sites where proteins are being made. These proteins are being packaged in the Golgi apparatus. And so as we go further into this, you'll see that this is a beautiful story that is about to unravel. We're setting ourselves up to find out what a healthy cell looks like so that we can start exploring what an unhealthy cell looks like, or a pathological state. This is what you will be presented with as a healthcare professional. You will see a person that has some type of condition or clinical symptoms, and those clinical symptoms can be drawn back to organ systems or even the tissues or cells that make up those organ systems. So it's important to think holistically as a healthcare provider. And hopefully this lecture is beneficial to you and you can answer some questions that have to deal with this topic. If you have any further questions, feel free to contact me at theRuffinNeuralab.com. Until next time, thank you.